Okay. So Izmir is the, the third, the biggest city in the Turkey. So we have got almost 900 beds capacity, and there is special departments like transplantation units, organ transplantation. So we have got a tissue typing lab. Now I am working there. And so we have got a multivisceral transplantation, liver transplantation, and kidney transplantation. So this program is really one of the biggest, the unit in this region. So we are doing the related tests and basically HLA testing and the PRA testing and cross-matched by using the flow cytometry, Luminex, whatsoever. And so during this COVID pandemic, this hospital at the same time is the pandemic hospital. So all the cases are coming through this hospital, and there are all the tests and all the procedures is going on here. So this is the reason we have got a lots of data, the patient's recording data. So, in future, in order to get the publications or scientific work, just the people are working on it. There is a committee for it. The Education COVID-19 Committee. They are organizing the projects. While we are doing so, we have realized that you have got a project in, the COVID and HLA related. So we wanted to join it. Our story started with that, like that. So we are really enjoyed very much. So at the same time, we have got a big data so far. Lots of COVID-19 cases and all the lab findings and radiologic findings, all of them we have got. And during the time, we are going to sort it out, all of them. Meantime, at the same time, in this region, there are universities. There are quite lots of universities. And these universities has got a lot of PhD students. Some of these PhD students works on the tissue engineering and cell therapy at the same times, the HLA, the immunogenetics. So, and some sort of projects just going on with this area as well. And this is the brief description of what we have doing so far. The second things, as you group the 4 groups, what we have done, related to your project, I think it is quite big project as far as I imagine. What we have done so far, just group 1, group 2, group 3, and group 4. From intensive care units, infection units, and labs are participated in this area. So the samples just coming through the microbiology lab. These samples with the hemogram tubes, and DNA is going to be purified. And later, according to your procedures, we are going to ship them, or we are going to work them. I don't know. Today we are going to know it. And meantime, I would like to say that our lab, FA accredited histocompatibility lab. We have been thinking that if it is possible, we would like to work the samples in our lab, or our lab may serve your project. We can participate in this area. I don't know what is your procedures, but as I said, we are going to learn. And I would like to stop in here, and I would like to give the microphone to you Again, I would like to say thank you for sharing this scientific, the data and making the very big projects in future, Seiamak. Okay.
Thank you, Ibrahim. And do you hear me okay?
Yes, I can hear you. There is no problem. Just in our conference room, there are our colleagues, and they are going to ask questions after your talk. And possibly you may not see them, but you can hear their questions.
Okay.
Okay.
So first of all, thank you for having organized this call, and thanks for all your colleagues to be around you. So I will briefly explain our work on COVID. So first of all, we are a university hospital in east of France, Strasbourg. It's a 2,500 bed hospital. And during March, April, the region was pretty badly hit with COVID. So for instance, at the height of the epidemic in the region, we went up to 200 ICU beds and approximately 400 regular beds only for COVID. So it was a very bad situation, but on a scientific level, most of the physicians who were taking care of COVID are affiliated to my lab. My lab main interest has several interests. One of them is genetics of the HLA. The others, which are more recent, are genetics of primary immunodeficiency syndromes and basically genetics of immune-related diseases. So we have our own whole exome and whole genome sequencing platform in the lab, so we can do a lot of things very, very quickly. And with respect to transplantation, I am the head of a national program to identify new histocompatibility antigens, and most French transplant programs, whether it's solid organ or hematopoietic stem cell transplant program, are part of this so-called laboratory of excellence. So getting back to COVID, like everyone, we were surprised by this epidemic. We focused on two different things. We always focused on patients who are under 50 with no comorbidities. With no comorbidities, I mean except being overweight, because that's something you cannot readily exclude. But without that, we started to studying patients under 50 with no comorbidities, and we are basically finishing the work on whole genome sequencing, RNA-seq analysis, proteomics, cytokines, etc., in a cohort of ICU patients under 50, no comorbidity, versus patients in a regular ward only with additional oxygen. So that's one part of the work, and the part of the work we will be discussing today is basically the HLA association with COVID, which is a unique opportunity to study HLA disease association, because by definition, this is a new disease, the new virus. So it's not like malaria or other microbes where there has been selections over generations. So this is a new virus. It's a bit like HIV when it hits a population. So it's an opportunity to see what HLA alleles correlate with different disease states. So it's an international consortium under the auspices of the International Workshop. All major EFI is part of it. ASHI in some part is part of it. And the first set of conditions that we set up, I realized that they are very drastic, because under 50, no comorbidities, so the numbers are very low. But let's say this is the pure cohort. And so that's one part. And since I have basically disseminated the first, the call for samples, we have gone, we have become more realistic in including all COVID patients, irrespective of age and comorbidities. So we will make two different cohorts, one all-inclusive, every COVID individual, of course, divided by these 4 groups, 1, 2, 3, 4, and then a very select and much more pure cohort, which is the original cohort under 50, under 50 with no comorbidities. So where are we at right now? In Strasbourg, we have had 700 COVID patients which were biobanked at our hospital. So we are right now extracting the DNA, and hopefully during the month of October, we will do the entire HLA, KIR, and MIG typing. And we are receiving samples from all over the world, some small, some larger. And hopefully the idea is by the end of the year, by end of December, reach around 5,000 patients, all-inclusive patients. And within those, then we do a separate study on the ones which are under 50 and no comorbidities. So, so this is in general terms the project. Of course, it's part of the next International Histocompatibility Workshop, but that's in 2022. So we cannot wait until that time to publish, because we are not the only one who are doing this. There are several groups in the US who are doing whole genome sequencing, and they can basically extract the HLA data from whole genome sequence. So we shouldn't be very confident that we will be the first. So we have to really, the idea is by the end of the year, having a manuscript ready. So yeah, that's in a few words where the overall picture.
Okay, excellent. As far as now, what's while you are talking, I have been thinking that we have got all-inclusive patients, we have got a lot. I mean, without making any classifications according to the age or comorbids. So in future, if this data is going to be important, we can provide this data by taking the permission our head of physicians or Ministry of Health. So you are saying that 5,000 now patients, all inclusive group.
Yes.
So it is quite big and a large group. So I think that we may have 1,000 possibly. This is not a certain number, I guess, but I can write it down to you. All inclusive patients, we have got a lot as well. So what we have done, according to these 4 groups, we grouped, and almost each group we have got almost 40 or 50 patients. It is not too much, it seems now, but after we are looking at the data of the patient's recording system, because when we check through this patient's recording system, what you can see, you may see that, I mean, the final decision of the patients, because sometimes what is happening, the patients, you may put it in the group 3, but sometimes these patients may have the IRDS, and you are just shifting it to the group 4. So by sorting all these things, I do believe that we are going to reach the total almost 200 patients. So I am going to share all of you. And the second things, what I have understood, you are also looking at the KIR and the MICA as well. And I do believe that the size of the samples and the amount of the samples is going to be important by, I mean, the working on the different projects, let's say HLA, CUR, and MICA, or whatsoever. So if you have got a, I mean, a given procedures or given, I mean, the guidelines for this, we may share and we can prepare ourselves in order to get the, I mean, the enough amount of the samples and the correct classifications.
With regards to HLA, MIC, and KIR, we basically do the whole sequencing at once. So the same amount of DNA will be sufficient to do all the loci at once by next generation sequencing. So we don't do it separately. Yeah, I mean, as you mentioned, it would be very helpful for the project if you can if we can include 1,000 all-inclusive patients and these 200 patients in four groups.
Yes. And these are not a certain amounts. I am going to check them. I am going to speak to all related clinics, and later I am going to give a given, I mean, the amount of the samples to you, how much we have got. But in here, we have got two things. The first one, so I have already understood that the samples is going to be worked in your lab or in France, or how all the samples are going to come through the lab, and how the results is going to be declared.
Okay, so the sequencing will be done either in our lab in Strasbourg or by fee for service at DKMS in Germany.
Yeah.
But that would be, of course, people who submit their samples will get detailed results. So if you get, the only thing we ask collectively is that we do the publication, this joint publication, but it doesn't preclude that afterwards on your own samples you do what you want. But the first publication is a joint publication, but then of course the results belong to the people who send the samples.
Okay, yeah, I understand. And the other things is it possible if I say that we may, I mean, work our samples in our lab, or do we have to send all samples to your lab or to the Germany?
It has positive points and negative points. The thing is that if we do it in our lab or in Germany, we will cover the cost. If you do it in your lab, you have to cover the cost.
Okay.
And the advantage of doing it in a central lab is that every sample will go through the same procedure with the same kit, with the same results.
Okay.
And, yeah, but
okay. So the other thing is the deadline for sending the samples is the end of the December, or?
I would say the sooner the better.
Okay.
The sooner the better. But what we have to publish something with the end of the year. By the end of the year. Whether it's on 1,000 samples, it's better it is on 5,000 samples, but we cannot just, once we get data, unless there is nothing to report, which would be unlikely, we have to report it. Yeah.
Okay, Seiamak, also, are you going to need other features of the patients? Let's say we sent the DNA to you and just sequencing it for HLA.
Yeah, I will send you. We have a Excel file with minimum level information that we need. Basically, age. I mean, some minimal clinical information that we need to stratify the patients. but it's not a huge amount. Yes, but we need this before doing the sequencing.
Excellent. Now, I would like to now ask the audience if they have got some questions. I'm going to just transfer them to you. Is it okay? 5 or 10 minutes if you are available. Okay.
Yes.
Arkadaşlar, soru sormak isteyen var mı? Onu başhekimimiz de burada. Bu konuşmadan sonra o bizimle hani direkt ilgili şey. Tabii, evet. Biz bu projenin içindeyiz. Ben onu şey yaparım. Yani mesela Fransa şu anda bulunduğu hastanede o bölgede 5000 tane vakayı toplamışlar. Now, okay. Okay, Seiamak, now our colleagues just want to know that is there, I mean, the minimum level of the samples you are going to work or you are going to be collected in here. So what your expectation, let's say, from us, how many samples we may contribute to this project?
Okay, so, as many as you can. Yeah, I mean, if you still have I don't know how is the situation where you are in Turkey. Is the epidemic still very active, or?
It's active, yes. It is still active, and the numbers is increasing. So yes, and also as I said before, this the hospital we are working in is a pandemic hospital. So the samples are coming every day. So I don't think that we are going to have any problem of the sample size or I mean the patient size.
Yeah, I would say ideally for the under 50, if you can reach at least 200 per group, that would be good. And overall, basically, even if you have 3,000 patients, we take them. Yeah.
Okay. Okay. Now I am going to translate all of them to my colleagues. Yani her bir grup için ideal bizim istediğimiz 200 olabilir ama sağlayabileceğiniz rakamlar o söylediğiniz, ben 50'şer tane şu anda var demiştim, 200 tane ediyor. Bununla da katılabilirseniz olabilir. Pandemi devam ediyorsa yüksek sayıda gönderirseniz daha iyi olur diyor. O şeye yalnız konuşalım. Çıkmadan evvel o şeye katılamadınız. Biz bir karar almıştık. Onu da sizle paylaşalım. Onu da lütfen şey yaparsak Şükran Hocam. Now, now our colleagues is saying that we are all of them very, I mean, cautiously looking at the procedure. So each group is carefully organized. So my colleagues is saying that we may reach what you said. So there is going to be no problem.
Okay.
Okay. Basically, I think everything is clear. So Seiamak, by taking this opportunity, I would like to ask you again, 2 or 3 weeks later, I am going to write to you. I mean, if it is possible, for our students, if you can give the seminar, it will be nice if you are available. I mean,
yeah, it will be either me or my colleague Rafael.
Okay, hi.
Yeah.
Hi. Okay.
Okay.
So, it will be very nice. Okay, I am going to generally on Monday we have got a scientific education meetings, and
Okay.
mostly COVID related. And I mean some Monday according to your availability, we can
Okay.
arrange the COVID related HLA or importance of the HLA in COVID immunology whatsoever. This sort of the titles, it would be nice. So we would be very grateful if you arrange this sort of meeting.
Sure. Okay. Okay.
Okay.
Okay.
So, okay, I would like to finish it in here. It was great to meet you, and we are really happy to participate in this project. I hope we are going to get very nice scientific results, and everybody is going to benefit for it.
Of course. Yeah, that would be nice. Yes.
Okay. Okay. See you then. Bye bye.
Okay. Thank you.
Bye-bye.
Thank you, Ibrahim.
Okay, bye bye.
Bye bye.
Kapatalım. Arkadaşlar öncelikle